for joining us. We're going to let everyone join in here and get settled, and then we'll get started in just a moment. All right, well, hello everyone, and welcome to today's webinar titled Elevating Cancer Research Workflows Embracing the Digital Lab Movement. This webinar is a part of the 11th annual event in the Cancer Research and Immuno-Oncology Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by eLabNext. For more information about eLabNext, go to www.elabnext.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located on the right of your screen. Please take a moment to answer the survey questions. It will be available during both the live session and the on-demand period. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located at the top of your screen within the event. All right, let's get started. I'd like to now present today's speaker, Margriet Mestemaker, Key Account Manager, Lab Digitalization Specialist at eLabNext. For a complete biography on our speaker, please find it on the left of your screen. Margriet, you may now begin your presentation.
Thank you, Kaylee, for your introduction. And hello all, welcome to this webinar. Happy to have you here. Let's dive in. So as Kaylee already said, my name is Margriet. And as you maybe can hear at the name, I'm a Dutchie, so I live in the Netherlands. Just a small bio, but I've earned my Master of Biomedical Science at the University of Groningen, and I specialized in cancer cell biology here. And at eLabNext, I of course enjoy educating researchers working in a life science lab on all the possibilities of lab digitization, helping them and maybe you, of course, to find the best possible solution. So let's dive into the agenda for this talk of today. First, we will look shortly at the future of cancer research. Then we will move over to why you should even consider or start using a digital lab platform or DLP in your cancer research. Then we will dive into some important functionalities of a digital lab platform. Then we will look into how you can future-proof your lab. And then, of course, I will guide you through how you can choose the right digital lab platform for your lab. So to give you some insights on how to do so. So that's what we will be discussing today. And of course, we will close off this session with a Q&A so you can ask all the questions that you have. So just enter them in the chat. So let's start. The future of cancer research. Probably already heard it a lot, but some of the hot topics, of course, in cancer research are also some hot topics in precision medicine. So the latest hot topics are the dynamic monitoring of cancers, for instance, but also the precision immuno-oncology, antibody drug conjugates as well, and also to try to see how the mutant-specific molecules and mechanics work. So these topics really thrive right now into the cancer research. And if we're looking to the other picture that I have, some other hot topics related to cancer research are also the hot topics in the immunotherapies. So for instance, combinations of agents that can target independent pathway or maybe combined pathways as well. Advancing effective cell therapies for solid tumors to how can we get rid of those, and also the drug target inhibitory pathways. How does it work exactly? And of course, nonetheless, or not less important than that, developing more effective cancer vaccines, as that's still a problem of today. If we are looking at those two are like the main topics in precision medicine and the immunotherapies, we all see that there's something that keeps them all together. And that's that we need all kinds of data, for instance, clinical data, but also laboratory data, research data. We need to convert those to knowledge and then, of course, to actions for selection of therapies, etc. But what everything combines is that all these types of data right now are somewhat not connected. We call them data silos, so everything is uh, sort of standalone. And that's why we need to ask ourselves, what do we need to make sure um, to connect those, to get to those to the path, to the knowledge and the actions, of course. First, before we go dive into that, before we do that, I will guide you all through a little bit of a history. So the history of the lab operational systems, and I will combine or compare them to your telephones. So in this case, if we are looking at 1861, then the first cable landline was established. And if we compare it to, for instance, the way that we track our research data, then we can compare it to the physical notebooks, the paper notebooks that are, of course, as old as science. If we dive deeper into the timeline, then we can see that in 1996, there is already a transition from analog to digital. So we are getting mobile phones, for instance, and also the first electronic lab notebook was established in 1994. And then if we go further into time, we are right now at 2023. We see that we all have smartphones, everything is digital. We are already used to that since 2007. And of course, we are not going to get rid of it anymore. And if we look at the place where researchers still store their data, we can see that that's still somewhat divided. So sometimes still paper lab notebooks, sometimes it's already a little bit digital, but what researchers really currently need is a lab operating system. So combining those data silos to get everything out of your data. So that brings us with at the question why you should even try to consider a digital lab platform for your cancer research. If we translate this to your cancer research, we can visualize the current state of research data as a multiple disconnected systems and processes. So at the left, we can see that you'll, of course, have all the data collection and storage that you have. So your instrument generated data, your paper lab notebooks maybe, or even a Word document or something that you're using, data on internal servers, something in between the data analysis and the data collection is all your omics data, which is of course thriving in the cancer research. We need to all do this and combine this data together. And that's something in between. If we are looking into further data analysis, we can see that you're maybe using GraphPad, Office Online, or some other research and productivity tools, but also maybe use some automated analyzers for that. Then on the bottom left, we can also see that next to the data collection and storage and the data analysis, we also have the inventory operation. So what chemicals are you using? So how is this tracked? Are these paper inventories stuck on freezer doors? or are these various Excel tabs that you're using, maybe something else or something delivered to you by your company, but usually this is also still separate from your data collection and your data analysis. And what we also see is that you also need to adhere to some compliance and research integrity, of course, which can be, for instance, SOP instructions that sometimes still are on paper or on PDF. And as you can see, this all is not managed in a proper way. We can see, yeah, we can locate all these different data silos, as we call it. So you have your data collection, your data analysis, inventory operations, and compliance, and it's all stored in different ways. So what do we need? And what the future, and of course also your digitalization need, is a seamless, adaptable, and integrated experience. As we call it, a digital lab platform, where all your data silos will be connected. So we call it a digital lab platform, where you can, of course, collect and store your data, but also analyze your data. You should be able to collaborate on those. So it should not be only you who's working on it, but also your team members, your co-workers, maybe your PI, lab technicians, other scientists. You need to work together and collaborate to get the most out of the data. And of course, you need to be able to identify and track everything that you have. So for instance, your inventory, but also the unique identifiers in your for your electronic lab notebook, for instance. So all this needs to work together. And of course, as every research is different, we all have some things in common, but usually every research is different, every organization is different. So you also need to be able to customize your solution and your platform. Looking into the possibilities and functionalities of a DLP, because now we looked into why we would need a DLP, why should we connect those isolated data silos, and now we were going to look into the possibilities and the functionalities of a digital lab platform. And I want to do this linked to the FAIR data principle. I can imagine that this is something that you already have heard of, or maybe it's new, so I will quickly show what I mean with FAIR. So FAIR data is about findable data, for instance. So the F is for findable. The data and the metadata should have a unique and persistent identifier to make them easy to find and also index, so it should also be searchable. This means that everything you put into a platform should also be able to get it out on an easy way. The A for fair data stands, of course, for accessible. So data and metadata should be deposited in a trusted repository that can be accessible to both humans and machines. So you can imagine that it means that you should be able to get your data out. You should be able to get to your data, but also that your data will be accessible only for the right people with permissions, etc. The I in FAIR stands for interoperable. So this means that the metadata should use a formal, accessible, shared, and broadly applicable language for knowledge representation to allow for instance, data integration, but also to make sure that the platform that you're using is also not a data silo on its own. And of course, FAIR, the R in FAIR stands for reusable. So everything you do you should be able to reuse it again. So data, metadata should have a clear usage license. Provide accurate information on provenance, enable them to be repurposed by machines or even humans. But those are the principles of FAIR that I would like to you to keep in mind during this talk, because we are going to connect those with with the possibilities and functionalities of a digital lab platform. So if we are looking into these possibilities, and the first thing that of course comes in mind if I'm looking at Findable, if you're looking at finding your data, or maybe this is a view that you'll recognize from your freezers that, yeah, if you're opening them, you don't really know what is where, how to find it. You see all those sticky notes and all the ice stacking up. This is not really findable, right? So if we are looking at Findable, what you would really need is a place where you can search for anything, any sample, for instance, that you would need. In this case, you can do an advanced search. You can even do a search within your group or within your organization. So this choice already makes it more easy to see if you're looking for something within your lab or maybe within your whole organization. You can also toggle a multi-line search. So if you're searching for something, it should be important that you are not limited in only the one thing that you enter, right? You should be able to find multiple things in one search query. I think that's really convenient to have. And just to show you how it would look like, we're now searching for pack 51. And then you should get a search hit, of course. And in this case, you should also be able to export to a usable format, for instance, CXV. So you can also do something with the data that you're finding in this case. Something that's also very important is that you should be able to save your search queries. Because if it's something that you're searching for a lot, for instance, if you're searching by expiration date and you want to have a list of all the samples that are expiring this month, you don't want to fill in the same parameters over and over again. You just want to be able to call up this query and to export your results of this month. So also it's findable, but in this case, if you click Save Search, it's also reusable. Next to, of course, that you should be able to search for things behind your computer, you should also be able to find things if you're in the lab. So that's why an integration with certain label printers and also the possibility to customize your label so you can see what's really on your tube, your box, your freezer, etc. That's really important. It's a really important functionality of a digital lab platform. So you can really customize your use and of course the findability features of a digital platform. Next to the freezers and maybe the look or the freezers that we all know, we also sometimes forget maybe where we put things, especially when life is busy. You might forget where you put your data if you're not used to working with a digital lab platform. So this might also be familiar to you. If it is, let me know in the chat because I'm always keen on hearing your experiences with that as well. So Findable is not, of course, only about finding your samples, but also about finding your data, finding your experiment results, etc. So that's also what a digital lab platform can offer to you. So in this case, we can see an overview of some projects that I'm working on in my demo environment. And as you can see, we can also search in this case for HeLa and PAC51. So we're in the same context in this case. And you can see that the two experiments are popping up. If I change my search query, because now it's searching for HeLa and PAC51, if I change my search, then you can see that also the results are different. So your digital lab platform should perform or multiple ways how you can do your searches, etc. And in this case, you can really see that it can help you find the results that you need or find the data that you need. What's also important next to finding your data is, of course, linking your data to maybe already existing references, for instance, by using something called Mendeley or another citation help like RefWorks or something like that. And you should be able to use it within your daily life. So in this case, the Mendeley you can use within your experiment, so you can directly create a durable link with the references that you're referring to. And if we're looking at, for instance, the scan to ELN, you can adapt with the scanner, so you can easily add, for instance, your samples by using the barcode scanners to your experiment. So it's really an optimized workflow like this. But is finding all your data enough? I know it's very important, but what about consistency of your data? So if we're diving further into the possibilities and functionalities of a digital lab platform, you can imagine that if you are working on some experiments, you're doing a lot of the same work, or you maybe have already some workflows in place, that you don't want to do the configuration exercise over and over again. So a digital lab platform should have something in place to accommodate that. So in this case, it is possible to create a study template, for instance. So this is my example of a study template, which means that you can link different experiment layouts, sort of templates, to each other, which can help you with the whole workflow. So for instance, the sample prep, your facts template in this case, your colony forming assay, and maybe you're even adding more to this. So you're not limited to 3, but just to keep it easy for you right now. So your DLP should really let you customize your experimental workflows. In this case, you can create templates from, for instance, existing experiments. So if you already worked on an experiment and you think, hey, I did a really great job, I want to repeat that, or I want to really copy this, then you can use that as well. But it is also allowing you to start new, or indeed to edit your template group, which links all your templates, all your experimental templates together. How does this look like? So in this case, we're working with different sections. So a part of an introduction, of course, maybe you can link your specimens and materials. So what chemicals did you use for your experiments? You can link them also with the scanner if you would like. And of course, you can add new samples, but you can also browse for existing samples. So this is really up to you on what place of the experiment you are at the moment. Of course, we also have our method, our standard operational procedures, or SOPs. So you can also easily implement them, and of course you can restart them if you need that. Need to restart the experiment, for instance. You can edit it, or you can turn the edit button off if it's not compliant with your regulatory affairs. And of course, you should be able to link all your data. So you can link your raw data files, for instance, or you can do some analyzing within your experiments. What just showed up in the screen now is that within the rules and permissions that you can set up within your digital lab platform, you have the possibility to see or to tick the box that you would like the protocols to be witness signed. So for instance, that you're only allowed to use approved protocols. So you can really see this as a flexible way of working, and within this flexible way of working, you can have some approved protocols that you can only use, for instance, to streamline your workflows. And next to the protocol signing, we also have our full customizable signature workflows in place. So in this case, it should be possible to really set a signing workflow for your project. In this case, for instance, a signature one, any collaborator, that the second signature should be someone with authority or with the permissions to do so, and maybe even a third signature if that's needed. So you can really customize your signature workflows that really matches your daily work and also the type of organization that you're in. Maybe you need to be working under GXP compliancing, so then this should be really helpful. So you should really look into the options for the signing and a witness signing for your digital lab platform. And now, with the standard operating procedures, with the signature workflows, you have managed to obtain your consistent data. What about access and traceability? Should anyone just have access to everything? Should some things be hidden or only be accessible for certain members of the team? That's all something you should think about while assessing for a digital lab platform. So we're just diving further into the possibilities and functionalities of a digital lab platform. And in this case, the accessible, we already talked a little bit about it in the beginning, is all about the A of fair data. So this is also one of the parts that I would like to highlight a little more. It means that your data and metadata should be deposited in a trusted repository, and it should be accessible to both humans and machines. Although this is quite difficult to answer in one way, there are certain certifications for digital lab platform vendors, for instance, that can really help you with assessing this part. One of these is the ISO 27001 certification for, it's the ISO certification for the data security, which a vendor of a digital lab platform should have, which ensures you that your data is safe. So if we're going to the next slide, you can see that, for instance, we are ISO certified for the data security, but there are also some other certificates or guidelines that a digital lab platform should adhere to. So for instance, a digital lab platform should be compliant with 21 CFR Part 11. So that's all about their traceability. Can anyone access anything? Any changes, are those logged or not? Etc. So same goes for the GXP compliance. So your digital lab platform should support GXP compliance. This is a good highlighting that it should support it, because usually a digital lab platform itself cannot be GLP or DXP compliant. We can only assist you to be DXP compliant yourself, because we are not doing the research, we are only providing the platform, but the platform should support it. So it's a really good thing to keep that in mind. And of course, nonetheless, or not less important, is the GDPR compliant. So as a vendor, we should be GDPR compliant, especially within the EU. And of course, we also have here listed the compliancy with the HIPAA or health insurance Portability Accountability Act within the US. And that's also something, one of the, I think, certificates, compliances that you should look into if you're looking into using a digital lab platform. Now a little bit more about customizable roles and permission, because we're talking about accessibility, and hand in hand that goes with roles and permission. So in this case, I just show you a preview of all the roles and permissions that can, for instance, be set up. And I think it's important to know that in this case, the roles and permissions are customizable, but are leading for the whole platform. So if you are not allowed to, for instance, add a compartment, then you cannot do it in any way. Also not in other ways, you can just not do it. I think what's very good to highlight is that there might be some differences in certain parts. So what is good to know is that the permissions within this digital lab platform usually are about if you're allowed to add something, delete something, update it, edit it, view it, or maybe move it as well. But for your experiments, for instance, there is a difference between your own experiments and others. As you can imagine, that can become very handy with the setup of your rules and permissions. And if you have set up the rules and permissions within this group, then of course you can distribute the rules very easily just by clicking on the user role and the user and edit it. Of course, you can only change this if you have the permission to do so, or if you are an administrator in this case. We're talking about accessibility. So we're talking about trusted repositories, but also the GXP compliance, 21 CFR Part 11, etc. Something that you cannot miss about that is the availability of a full audit trail. So in this case, it's important to have a full audit trail of all the parts of your digital lab notebooks, which can be, for instance, experiments, or maybe even parts of experiments. We call those sections, or even your attachments, your protocols as well, samples, sample series, storage units and compartments as well, equipment, permissions, login, user activity, and maybe even more in the future. Just to highlight how this audit trail is displayed, in this case, you can see an audit trail for a sample. The name is changed during the creation, and then you can see that it's updated. The parent is cleared one day. Let me see, the location is changed, and everything is tracked on a user base. In this case, it's all me because I worked on this, but just for you, to give you an idea that it's really tracked on a user level, which complies fully with all the GXP compliances, etc. So we talked about what we can do now, what is important. We talked about accessibility, findability, things hopefully you can all relate to. But what about the future? Unfortunately, we don't know what the future holds. Yeah, maybe it's even better that we don't know, but we just don't know. So what should you be looking for in a digital lab platform? That's something to future-proof your lab. So it should really be future-proof. One thing you can look at is are the customizability options. So can you customize your experience? In this case, it grants you limitless possibilities. All the data that you put into your digital lab platform is exportable into PDF, but also XML, HTML, and JSON. So you can really do still something with your data. It's not only in PDF. You can also personalize beyond what is provided by your software vendor. So there are many options that you can change your experience. And of course, you can have buy something extra. For instance, hey, here is it listed extra cost based on your lab's budget. So if you have the budget and you want something special, we provide the tools that you can make it. So that's also why it's linked to future-proofing your lab, because with our platform, we allow third-party integrations to be a part of your experience. So you can really integrate those other platforms, for instance. Some are already by default into our platform, some maybe not. We also do our own developments, so we really are planning on staying on top of the line and future proof. But how does it look like? So at eLabNext, we know that every lab has its own specific workflows and that there's always different features that are important for some labs, but not for others. So for this reason, we provide an open ecosystem, which gives your users the possibility to personalize their ELN by integrating or customizing any functionality within the software. With help of our eLab API, users can customize their software by integrating third-party software applications. And those can then work with the data from eLabNext or the other way around. The API allows you also to retrieve data from eLabJournal and put it into processes within the third-party applications. Or of course, you can write data generated by other software systems to eLabJournal as well. So the possibilities here are endless. Additionally, we also offer users the ability to extend, integrate, or customize any of our eLabJournal features using our software development kit, or eLab SDK as we call it. Our software development kits are software tools and programs that developers can use to create applications. So this is mainly the place where all those actions that you ask with the eLab API can be combined. And for instance, you can generate buttons or you can add more features to really match the users at your own lab. So the API used together with the software development kit will really allow you to customize and extend the functionalities of eLabJournal accordingly to your demands of your workflows in this case. So what I already discussed about the eLab API and the software development kit, the third-party vendors were already mentioned. So we have what we call the eLab Marketplace. This is a place where you can find and install so-called add-ons, as we call it, or you can sort of compare it with the App Store on your phone. So you can install these add-ons and you can really set up the workflows, the environment that fits your daily work. A lot of these add-ons are just for you to use for free. Some are used by third-party vendors, and some are asked for a small fee on top of the license fee. But it's all transparent and all up to you what you would like to use or not. And of course, with the help of the eLab SDK, you can create your own so-called add-on in this marketplace as well. So if we're going to look into some of those add-ons, just to give you a small idea of what the possibilities are, one of those is the option to link equipment and generate results within your experiments directly. So we have a plug-and-play add-on with, for instance, Implan, the nanophotometer. If you have a reading, you can directly let it be updated to your samples, for instance. Another possibility of future-proofing your lab is the integration with Cluster Market. This is an equipment scheduler. Maybe you already have heard of it. And in this case, you can really link the data that you put into Cluster Market into your electronic lab notebook. If you schedule within Cluster Market, you can also directly link it to your experiment, so you can see when the experiment was performed, etc., at what time slot, and so on. Another integration that we are very proud of and that's used a lot within our current users is, for instance, the Office Online server or the Office Online integration, which allows you to use Excel, PowerPoint, and Word Online directly into your ELN, including a preview of all your results. And not to miss is our own add-on called eLabWebEdit, which allows you to open any file type with your computer. So your computer is searching for the right program to open, make changes, save it without having to download and upload again into your eLab journal, which makes it very easy to integrate all kinds of annotation tools or all your data as well, so you can visualize it easily. But of course, there are many more options there as well. Um, Also nice to know is that, of course, the power of AI and machine learning is also taking over the world of, I think, cancer research as well, but also with the digital lab platforms. So we also have some add-ons in place for that, just to highlight the options with, for instance, Rewire Neuro, that you can really make the most out of your data, so you do not have to do the analysis by yourself, but you can use AI and machine learning for that as well. And the same goes with, for instance, Immunomind, also an add-on in our marketplace, where you can really easily use the tools of AI and machine learning for all your data analysis. So now we already talked a lot about why you would need a digital lab platform. What are the basic functionalities and what do you really need and why do you need them? So now let's look at how you can choose the right one for your research. So let's go back to FAIR. If you go back to FAIR, about findable, accessible, interoperable, and reusable, they all kind of came along in this during this webinar. We also know that FAIR is faster. So in the end, it can save you time. But as we also can see is that already the first papers are being released. In this case, also together with us to really show how you can benefit from connective software. So if you want to know more about it, you can also find this on the internet. But now, to answer that, how can you choose the right digital lab platform for your research? And I think what is key is that you need the customization to suit your needs. So in this case, you should really look for a platform that has something in place, like the eLab API, so the application programming interface that allows you to be the boss of your own data, to make sure that you can really customize the input and the output of all your data. Some of the researchers that already went through this process before you, I just shown them here, and to give a quick overview of what some customization can look like, just to show it here. This is a dashboard made by one of our users in Power BI, and they just created a dashboard. So with the API calls, they got the data out of the Digital Lab platform, created a custom dashboard for everything they needed to know, and then created this dashboard so they can work easily. So it's really a place where you can get the most out of your data. So if we go back to the challenges in cancer research to link the data together, to move away from those isolated silos, we really know, hopefully now, that you really need some place where you can integrate all this together to do even more with all the data than you currently might even imagine that it's possible. So really focus on what type of data you have currently, and also how you can visualize it and what you need. So if you have those key points, then you would really know what type of digital lab platform would suit your needs. So if we go to what steps you need to perform, how can you select the best lab solution for your lab, whether it's a system with an open ecosystem or not, how can you even start? I think that's a great question to answer now. So first of all, you can ask for the availability of APIs and software development kits. You should identify your key users, so you should know whom you need to ask, but also who knows more about your research than you. So really use that. You should identify your business needs and ask questions, of course. Then you should seek lab digitalization experts and really try to find out how this platform can elevate your research. And how you can do that is just to schedule a discovery call. Just get to know the vendor, but also let them to get to know you, so you can get into touch and really see if the platform would be a fit for you. Of course, then you should schedule a personal demo. So don't stop with the discovery call. Really get a personalized demo where they can also show you what they can do with your data. What can the platform do? How can they elevate your research and experience? And of course, then after that, you should be able to get a free trial of the platform so you can try it along. So start your free 30-day trial. And see how it goes. So that's kind of my talk for today. Really happy to have you here. Thank you all. And I think it's time for the Q&A.
Yes, it is. And thank you so much, Margriet, for your informative presentation. So we will now start the live Q&A portion of the webinar. To our audience, if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. All right, let's get started. So we've already got some great questions coming in from the audience. And our first question asks, what is the primary advantage of using the eLab API to customize eLab Journal?
I'm sorry, something's happening with the slides, but they are just moving along. But we can have it on this screen. That's not a problem. Kate, could you repeat the question, please?
Yes, of course. This question asks, what is the primary advantage of using the eLab API to customize eLab Journal?
Thank you. Yeah, the primary advantage of the eLab APIs is that anything you can do on the front end, so what you could just see me showing to you, you can also automate this. So I think that's the most important thing about it, that you can automate your research workflows on the places that you really need it. So you're really flexible in working with this customization.
Great, thank you. Another question we have here, can the eLab API be used independently of the eLab SDK?
Thank you. Yes, definitely. That's a very good question, because we always show them together, but they can definitely be used next to each other. So you can really use the API calls already also from our browser. So we have a website set up for that, so you can really use the calls without to do any programming yourself, and that can be done without the software development kit, so without the eLab SDK. The eLab SDK cannot be used without the eLab API because you need those to brace them together, to bring them together.
Great. And it looks like we have time for a few more questions here before we wrap up, so we'll continue. This next question asks, can eLab Journal be configured to establish a quality management system?
That's a very nice question. Yes, it's that's definitely possible. I think it's good to emphasize that eLab Journal itself is not a quality management system, but you can really use it to set up your the quality management systems as well. With the signature workflows, for instance, everything you set up there can be used to really link to a quality management system. And if you would really need to, you can create an integration with your quality management system with help of the API and software development kit.
Wonderful, thank you. Now we have time for one more question here, so we'll wrap up after this one. And this question says, our IT has strict rules about new software and data security. How do you address those concerns?
Something else than the eLab API. That's very nice. If your organization has very strict compliancy rules, then we can support that. Maybe I can go to the slide where it was moving to before, just to show you that we offer different hosting solutions. So eLab Journal is a cloud-based solution, and you're using it by using your web browser. And if needed, you could have it installed on your own servers. So if you need to get all the security in your own hand, then it's definitely possible to install it on your own servers. We've done it before, and we can do that, of course. And next to that, we as a company are ISO certified for the data security. So those together should really be compliant with your security concerns.
Great. Well, thank you so much, Margriet. This does bring us to the end of our session today. Do you have any final comments for our audience?
Thank you. Maybe just to let everyone know, go back to the last slide to check and try yourself. Check your digital lab platform. Try it yourself with a free trial. If you're happy, I can reach out to you with that. And to close it with that, thank you all for listening. Have a great day, and maybe we'll see each other again later.
Absolutely. Well, thank you again, Margriet, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, eLabNext, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until October 7th, 2025. Labrets will alert you via email when it's available for replay, so we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.